Okay, I think we're recording. Hi everyone. Today we'll be talking about Hurler's disease for biochem, short for biochemistry. So here are the objectives that we will cover today, and we just took these objectives straight off the syllabus. So I'll be covering the first two. So what is Hurler's disease? It is a rare genetic metabolic disorder, more specifically a lysosomal storage disorder. And there's more than one types of these metabolic lysosomal storage disorders. And these are all part of the mucopolysaccharidosis family. And mainly this disease is caused by a deficiency of an enzyme, which we will talk more about later, which leads to an accumulation of glycose aminoglycans in lysosomes. Well, what is a mucopolysaccharide? These are also known as GAGs. And all you need to know are that they are highly polar polysaccharides that exist within the connective tissue, such as bone cartilage, connective tissue on heart valves, etc. And when they are not disposed of properly by the lysosome, they cause defects on these tissues, causing malfunctions. Well, I know what a lysosome is. We learned in general biology that the lysosome is the cell stomach or recycling center using enzymes to decompose waste. And without a specific enzyme, a lot of cell waste can build up. And the trash in this case, which are GAGs, are long chains of sugar molecules. And these can't be broken down easily. And when they build up, they cause the cells to swell up and the cells malfunction. And then here are some basic symptoms of Hurler's disease. The main one is corneal cloudy, which is the most outer layer of your eye. And there are mucopolysaccharides in the cornea. And when they are not controlled properly, they can have ocular manifestations. Another thing is coarse facial features. These GAGs build up and thicken the skin and change the bone structure, leading to large forehead, flat bridge, enlarged tongue, joint stiffness, and other cardiac and hepatic diseases. And the picture on the right is from Cleveland Clinic. And next, I'll hand it off to my colleague.
In this slide, I'll be talking about disease progression and diagnosis. So for some cases, this can be picked up through prenatal screening, like genetic testing, if family history is known. But babies usually look completely normal at birth. After about 12 to 24 months, physical symptoms start showing, like a change in facial features as well as other symptoms. Then developmental delays happen after around 24 months. This first affects speech and later affects hearing and sight. Along with other developmental changes, learning and sensory problems can also worsen over those few months. Hydrocephalus also develops after the age of 2. GAG levels are measured in urine to identify the MPC1 disease. And then enzyme activity is measured to specify the type of MPH1 subtypes, which identifies Hurler's syndrome. And this is usually measured through enzyme assays or DNA analysis. So in this slide, I'll be talking about transmission and susceptibility. Hurler syndrome is inherited through an autosomal recessive genetic pattern. This means that the infant must inherit the mutation from both parents, and the inheritance is independent from the sex. Patients who are more susceptible to inheriting Hurlers are when both parents are carriers and in populations where the parents are closely related. Patients with hurlers are not more susceptible to catching infections because this is not an immune disorder, but they're more vulnerable to specific medical complications because of GAG accumulation in the body, which could lead to heart issues as well as brain damage and airway problems. In this slide, I'll be talking about the epidemiology. So Hurler's disease is rare, affecting about 1 in 100,000 live births worldwide. When you include all forms of MPS1, including milder cases, the numbers range from about 1 in 50,000 to 1 in 150,000 live births, depending on the population. Hurler syndrome specifically accounts for the smallest proportion numerically, but is the most severe case. Untreated Hurler syndrome usually lead to death in early childhood before the age of 10, but with current treatment and more more treatment in the future, survival in adolescence and adulthood is increasingly common. Hurler's syndrome is shifting from a fatal childhood disease to more of a chronic condition.
And now we're going to talk about the molecular cause, like how does this actually form? Originally, this starts in a mutation in a certain gene, the IDUA gene, that encodes enzymes. The enzyme that actually you saw on a slide earlier when my colleague Jeff was talking. This is on chromosome number 4. And there's actually a lot of different mutations that can be found at this locus on this chromosome, which is why there's a certain family of syndromes and diseases. They're all formed from this mutation, whether it be Hurler or the other ones you see on screen.
Now getting to treatment about Hurler's disease. So one form of medication is enzyme replacement therapy, in which the alpha L iduronidase is injected weekly into the patients. Usually patients are injected with the enzyme replacement therapy in the earlier stages in order to prevent severe complications. However, the most effective form of treatment is hematopoietic stem cell transplant, otherwise known as a bone marrow transplant. Uh, this type of treatment clears a lot of the symptoms of Hurler's disease and is useful in preventing further disease progressions. Specifically, it has shown to improve the following, such as cerebrospinal fluid pressure, cardiac function, joint mobility, and hearing. As for preventative treatments, uh, there are preventative treatments for dealing with the complications of Hurler syndrome, some of which can include surgical procedures such as carpal tunnel release or cardiac valve replacement. However, a lot of these treatments can't be done properly because the afflicted individuals have sensitivity towards anesthetic surgery. In addition to there are other preventative treatments such as respiratory support, such as a CPAP, as well as hearing aids and gastrointestinal medication. And finally, uh, there have been animal models that have been used to show the viral vectors, uh, which have been used in transmitting the enzyme into animal models. Uh, these are all in vitro experiments as well. They've been shown improvements in liver, spleen, and brain function. Overall, current research is focused on gene therapy, such as Orchard Therapeutics and their Hercules program. Here, the figure on the left can see their timeline of study, which spans over 5 years. And one of the principal investigators, Dr. Maria Esther Bernardo, mentions, and I quote, previously reported results from an earlier proof of concept study in children shows robust metabolic correction, continued cognitive motor, and physical development, as well as early improvements in skeletal, ocular, and auditory health, showing that current research is taking great strides to improve on the Hurler's disease.
Being a disease that has such a high mortality rate at such a young age, the importance of early detection is really high for a disease like this. Because if you were to detect that you have this disease early on into its life cycle, you're allowed to intervene in its progression much earlier than you would have been otherwise. And alternatively, because it's a progressive disease, you're actually allowed to save a lot more of the body the earlier you detect it. So being a progressive disease, we've seen higher survival rates and significantly better outcomes for different organs in the body if you were to be treated earlier. Something like HSCT can actually help prevent this irreversible organ damage and the cognitive decline that comes with this disease. Assuming you were to detect this disease early enough that you can save a lot of the parts of the body that might be being attacked and in decline in this timeline. All in all, this disease, Hurler's disease, is a really rare disease, but it is very, very hard to deal with. It's oftentimes fatal and fatal at a young age. We've seen that there are different transplants and gene therapies that are in current development to help treat this disease. But all in all, right now, we're still in the works trying to find out what we can do about this disease and what more we can do in the future about it. Thank you.
Thank you.
Thank you.
Oh, and here are our references.